Assalamu alaikum, everyone. My name is Waad Hammad. I'm a second year medical student, and today I'm going to be talking about gastric physiology. I have my contacts here, so here's my number and here's my email if anyone wants to contact me. If you have any questions whatsoever, feel free to contact me. And if you want to stop me in between explaining, please do let me know. Okay, let's begin. Before I start, I just want to emphasize one really, really, really important thing is that, guys, Dr. Angelica's questions that she uploads on Moodles, the most important thing that you can do, honestly, like I cannot emphasize it on it enough. All our questions were very similar to the ones that she posted on Moodle, so please do make sure to do that. And without further ado, I'm just going to be talking about our objectives. First, we're going to be doing the stomach anatomy and the cast of cells. So what cells do we have in our stomach? Then we're going to go on to gastric acid secretion phases. What are the phases? Everything about hydrochloric acid. It's a lot. Surprisingly, you have to know a lot about hydrochloric acid. So I'll also be talking about that. And then last but not least, I'll be talking about gastric motility and emptying, inshallah. It should go all well. Okay. First objective, stomach anatomy and cells. Okay, so here we have our stomach. I'm just going to be breaking it down into different regions. So first we have our upper lower esophageal sphincter and our cardia, okay? And this area is where we have mucus and bicarbonate, and its motility is receptive relaxation and prevention of reflux. Now, you might have not understood anything, so let me just break that down right now. We have our lower esophageal sphincter, which is where I'm pointing it with my cursor. And then this area in specific is called the cardia, okay? And this area, the secretion that's mostly there is mucus and bicarbonate. Mucus is mainly for the protection of the stomach wall, and then we have bicarbonate, which is there to balance out the acid or to buffer it, right? And then for the two motilities, we have receptive relaxation. What is receptive relaxation? Receptive relaxation is when we have food coming down. Obviously, it needs to enter our stomach. So in order for that to happen, we need the lower esophageal sphincter to relax, and we also need the cardia to relax. This is so food can enter freely and it just doesn't get stuck there. The another use for the lower esophageal sphincter is prevention of reflux. And what reflux is when acid tries to go back up the esophagus. So we don't want that. It can cause a lot of damage, esophageal cancer. It can cause something called GERD, which is gastric esophageal reflux disease, which does a lot of bad stuff as well. So we just don't want that. Okay, so the second region of our stomach is the fundus and the body. And in the fundus in the body, we have secretion of a ton of stuff. Like you can see over here, there's a lot of things. We have protons, we have intrinsic factor, mucus, we have bicarbonate, and you can see two enzymes here, pepsinogens and lipase. So, and its motility is reservoir and tonic force. So we have acid, obviously, for digestion, and then we have intrinsic factor. I will be talking a lot more about intrinsic factor later on, but for now, what you need to know is that it helps you absorb vitamin B12. Then we have mucus and bicarbonate. Again, this is for protecting the stomach wall. Mucus provides the physical barrier, and bicarbonate acts as a buffer. And then pepsinogens and lipases are your enzymes, okay? And our motility here, reservoir and tonic force. Reservoir just means it's keeping food in an area. Obviously, when you eat, the stomach is where your food goes, so that's why it's in reservoir. And then tonic force is because it's always contracting, it's always doing movements to try to break down your food. And then last but not least, we have our antrum and our pylorus. Here again, we have mucus and bicarbonate, just like the lower esophageal sphincter and the cardia. So that's nothing new. Again, its function is purely protective. And then for the motility, it's mixing, grinding, and regulation of emptying. Okay, so again, I think this is all, mixing, breaking down, and regulation of how much food should go into the duodenum at once. We don't want all of it going at once and overwhelming it. So now that we know our stomach anatomy, we want to know what cells you can find in which regions. Now, I want to emphasize or begin by saying that I'm not saying that these cells can only be found in the body and fundus and the other cells can only be found in the antrum. No, it's sort of a mix, but what I'm trying to say here is it depends on the majority, right? So for our body and fundus, which you can see over here, we have the chief function, which is secretion of acid and digestion. So in the body and fundus, we have everything that has to do with breaking down food, everything that has to do with making it into smaller chunks so that it can safely enter our duodenum in this region. So we secrete acid, we secrete our hydrochloric acid, but we also secrete our enzymes, which breaks down the food. Okay. And in this region, we have our parietal or oxyntic cells, our chief or peptic cells, our enterochromaffin-like cells. Um, you don't have to know what that is right now. Don't worry, we're going to go deep into what the cells are. But just for now, just know these cells are in this region. And then we have our antrum, which also has three cells. And the main function for the antrum is actually regulation of acid and protection. So in our body and fundus, it was just secrete acid, secrete acid, secrete acid, and digest. But in our antrum, it's more of how much acid do we need? How should I increase the acid or should I decrease it? As well as protection. Because the food is just about to go into the duodenum when we have it in the antrum, we want to protect our duodenum from any acidic, any enzymes, anything like that. So we try to taper it down a little bit. We try to make it more alkaline, things like that. And then in our antrum, we have G cells, mucosal cells, and D cells. Again, you don't need to know what that is. I'm just saying that for now. And then again, I want to emphasize that mucosal cells and G cells can be found in the body as well. In fact, they are. It's just that they're more prevalent in the antrum. Now that we know our cast of cells, it's time to go deep into what each one of them are, what they secrete, what their function is, and what is their stimulus, or what is the reason they come out, right? So first cell type is we're going to talk about is parietal cell, which I drew over here. Now, parietal cell secretes two things, and they both have their own function. The first secretion that parietal cells have is hydrochloric acid. And hydrochloric acid is important for decreasing the pH, it's important for activating enzymes, all of those things. And it's also important for killing bacteria. Okay, so if we're afraid of an infection in our stomach or anything like that, hydrochloric acid is the agent we use to get rid of any of that fear, to get rid of bacterial infections, right? And then we also have intrinsic factor, which is also secreted by parietal cells, and that is also very important, and it helps in vitamin B12 absorption. Memorize this like your life depends on it, intrinsic factor for vitamin B12 absorption, because it's a very common question that they like to ask. And then for the stimulus, for what releases for what causes the parietal cells to release hydrochloric acid and intrinsic factor, it's ACH, gastrin, and histamine. You are going to see these stimuli so often, and I will get into it, but really quickly, I'll explain why of each one. So first of all, when you are hungry, you are in your rest and digest phase. You are in the I have the time to eat. I'm relaxed. I'm not thinking of, will I fight a bear in the next second? So this example, you may have heard it like a million times. It's very consistent with parasympathetic nervous system. So you're relaxed, everything is fine. And part of our parasympathetic nervous system is our vagus nerve. Our vagus nerve is it's responsible for allowing us to digest, for giving us food. So when we're relaxed, when we're thinking of digesting anything, our vagus nerve releases ACH or acetylcholine. And that is what stimulates parietal cells to be like, oh, she's thinking of eating, or she's about to eat, or she's relaxed. Let me release hydrochloric acid and intrinsic factor in advance. So when she does eat, it's going to be there already. Gastrin, you're going to see it in just a second. It's a secretion that one of the cells secretes, and histamine is also something else that is secreted by one of the cells. So we're just going to see that really quickly. Chief cells, now chief cells, I drew it in green over here. They are the enzyme cells, enzyme, enzyme, enzyme. They secrete pepsinogen and they also secrete gastric lipase. Pepsinogen for digesting proteins and gastric lipase for digesting fat. It's pretty straightforward. And then again, we have ACH, which stimulates it. Again, it's released from the vagus nerve. It helps stimulate it again because you're thinking of digesting. So your stomach is like, let me release enzymes in advance. Acid, if the pH is low, our chief cells are like, this is a perfect environment for me to secrete some enzymes. So let me do it because acid activates the enzymes. And then we have secretin, which again, its name is straightforward. It's a secretion that helps secrete chief cell enzymes. Okay, third cell that we have. Oh, before I go to the third cell, as a first year, I used to get very confused between parietal cells and chief cells. I used to always mix up the two of them. So a way that I would remember it is that chief sounds a little bit like chew, and chew is about digestion. So you can just remember it that chief cells chew digestion, so they release enzymes for digestion. Yeah. Now onto our, oops, sorry, onto our next cell, we have enterochromaffin-like cells, and I drew it in pink over here. Now, enterochromaffin-like cells releases histamine. Histamine is a cell signaling substance, and it acts locally, right? So it would go to our parietal cells and it activates it. That's its only job. It's basically its job is to stimulate our parietal cells, right? So we have histamine. It's when it's the right environment, when we have ACH released or when we have gastrin released, the histamine will go to our parietal cells and cause it to secrete gastric acid. So far, all of the three cells that we have in our body all are for secreting acid, getting or for digestion. Okay. And then we're going to move on to the the cells that we have in our antrum. Okay. Our first cell is called the G cell. And now this cell is very important. You might have seen gastrin quite a few times for their stimuli, and this is where gastrin comes from the G cells, okay? So gastrin is typically secreted into the blood, so it doesn't go directly, unlike enterochromaffin-like cells, which release histamine locally, so it can go through the stomach just to any surrounding parietal cells. Our G cells secrete into the blood, so just imagine the bloodstream is outside of the stomach here. And then when the time is right, it can stimulate our parietal cells. So this is something that also stimulates our parietal cells or even our enterochromaffin-like cells. It releases gastrin, goes to our enterochromaffin-like cells to release histamine to stimulate our parietal cells, or it can release gastrin and then it directly stimulates our parietal cells to release gastric acid. So either way, its job is just to make sure that the stimulation of releasing hydrochloric acid is there when it happens. And then our stimulus for it again is ACH. We want more acid. It's there by ACH, always, always, always. And then we also have peptides and amino acids. Peptides and amino acids stimulated is because when you eat, there are obviously peptides and amino acids in your stomach. So the G cells see that and they are like, oh, she ate. So they release gastrin into the bloodstream. Next cell we have is our mucous neck cell. Okay, now we're going less of let's secrete acid and we're going more to how can we protect our stomach? How can we make sure that nothing damages it? Our first thing that it secretes is mucus, and mucus acts as a physical barrier. I'm pretty sure you've seen mucus as a protective mechanism in so many places, but in the stomach, it's especially important that mucus is there because if it penetrates the walls, it can cause ulcers, it can cause a lot of pain, just so many bad stuff. So we need the mucus there. It also secretes bicarbonate, and bicarbonate is alkaline. And as you know, this the acid, the stomach is so acidic. So we do need bicarbonate to buffer the gastric acid. And it's it has tonic secretion, so that means that it's never specifically stimulated. Like we don't say our body doesn't say mucous next cell, you need to secrete mucus right now. It's just always being secreted because we always need that protection. But it can be increased by irritation. If our stomach is being very irritated, our mucous next cell can release more mucus. Okay. And last but not least, finally, we have our D cells, which are also found in the antrum. Our D cells secrete somatostatin. And what that is that it goes to our parietal cells and it makes sure that there's no more gastric acid secretion happening. Okay, enough is enough. This is when, in certain cases, for example, we have too much acid, like our pH in our stomach goes too low. And that's not good because if it goes too low, it might be able to penetrate the mucus barrier and get past the bicarbonate and then cause an ulcer. So our D cells detects the pH in our stomach and they're able to secrete somatostatin so that we don't have too much acid in our stomach. And again, its stimulus is acid in our stomach because it wants to stop that. And just one thing that I want to like a way for you to remember this more easily is that all secretions that are related to increased digestion, either it being hydrochloric acid or being enzymes, are all stimulated by ACH. So parietal cells, chief cells, enterochromaffin-like cells, and G cells all give you more secretion of acid or more secretion of enzymes, and they're all stimulated by ACH because digestion is all parasympathetic vagus nerve. Okay, so finally, we've gone through all of the cells in our body. Quick recap, parietal cell is HCl intrinsic factor, chief cell is enzymes, enterochromaffin-like cell is histamine, G cell is gastrin, mucosal cell is mucus, and D cell is somatostatin. Now that we know all of our cells, we're done with the very first section of our presentation, and now we have a checkpoint. If anyone has any questions, feel free to ask me. You can go ahead if you want. It will not have, but this is one of the questions, it will not have P7 and its intestinal response to meal and pancreatic secretions. This is just P5 and P6. And if everyone doesn't have a question, I will give you your first question over here, which is, which of the following factors have a direct action on parietal cell to stimulate acid secretion? And you have all of these options over here that you can choose from. I'll wait a bit for you guys to answer. Okay, I see you guys saying 3, which is correct. It is number 3. And the reason is because gastrin, we have all of the different types of secretions that could be secreted by a cell. And number 3 says gastrin, yes, which is correct. Gastrin acts on parietal cells and it stimulates it to release acid. And then somatostatin, no, because somatostatin would inhibit the acid secretion, so that's not an option. Acetylcholine, yes, because it's our common one. It activates almost every cell to release acid or to stimulate it in some way. And then histamine, yes, which is released by enterochromaffin-like cells. So good job, guys. Now we'll move on to the next part, which is gastric secretion phases. Now, this is a little bit heavy, so if you feel confused at any moment, feel free to stop me or ask any questions, and we'll get right into it. So our first phase is the cephalic phase. I think you guys already took a lecture on this, but I will still go through it just in case. So the cephalic phase accounts for 30% of the gastric juice in our body. And basically what it is that when you see food, or when you smell food, or when you even hatta when you're tasting food or you're imagining it, your body starts preparing for that because it knows somehow, subhanAllah, it knows that you're gonna eat food in the near future. So it's starting to prepare for that. So our vagus nerve picks that up and it's like, okay, she's gonna eat, we have to prepare the stomach. What's going on? So first, it will send ACH to our enterochromaffin-like cells. Okay? As we said again, enterochromaffin-like cells releases histamine. Histamine stimulates the parietal cells, and then so on and so forth. Same thing happens with the G cells. As again, we said the vagus nerve releases ACH. ACH will activate the G cells, and then G cells will release gastrin. So now we have both histamine and we have gastrin, and both of them will activate our parietal cells. And then also directly from the vagus nerve, ACH activates parietal cells. So we have histamine, ACH, and gastrin all working on the parietal cells because our body's like, you need to release acid, you need to be prepared. So our everything is working overtime. And then thus more HCl is being secreted. That is for our cephalic phase, which is just the preparation phase. Next, we go to also, one thing I want to add is that the vagus nerve also releases a different neurotransmitter called GRP. Okay, and this is a very important neurotransmitter because it's in addition to ACH. So ACH is not the only neurotransmitter that helps, but there's also GRP, which is very important because we have a clinical case that will relate to this. So just remember that our vagus nerve still releases GRP, which stimulates G cells, which causes it to release gastrin, which can then stimulate parietal cells. So GRP stimulates G cells only, not enterochromaffin-like cells, not parietal cells. But it still somehow increases HCl secretion because we have more gastrin. And now we're going to talk about something clinical. If we have a vagus nerve, so what a vagotomy is that it's complete removal of the vagus nerve. No more vagus nerve, bye-bye. This will completely abolish the cephalic phase of gastric secretion. Because as you can see, the vagus nerve is responsible for everything here. It releases ACH, it releases GRP. We have no vagus nerve, we have nothing. Like we cannot have it anymore because it's responsible for releasing ACH and GRP. So this is because stimuli that produce gastric secretion in the cephalic phase are all mediated primarily through the vagus nerve. All of them, it's either ACH or GRP. Without it, we cannot have the cephalic phase. So this is one thing that's important. Another thing that is important is that if we used a muscarinic blocker, and what the word muscarinic means is that it's a type of receptor, okay? And as we said, ACH mediates a lot of the functions in our body. So when we have a receptor and we have ACH, obviously ACH needs to bind to the receptor. But what happens if we have something blocking the receptor so that ACH can no longer bind? That is what we call a muscarinic receptor. And an example of a muscarinic blocker is called atropine, but this will not abolish the cephalic phase. Why? Because although we're blocking ACH from binding, we're not blocking GRP. So ACH will be blocked because its receptor is blocked, but GRP will still be there. So even though the gastric secretion will be a lot less, we still have GRP to stimulate our G cells, so our G cells can still release gastrin, and then gastrin can still stimulate parietal cells. And I basically just wrote that all into words here. Okay. I got a question. Would a vagotomy abolish HCl release in the gastric/intestinal phase? No, it would not, because we have other things that influence it. I will get into it right now for the gastric and intestinal phase. Okay. So now we're going to be looking at the gastric and intestinal phase, which is pretty long. So just stay with me, okay? For our gastric phase, it accounts for 60% of gastric juice, okay? So this is the big chunk. Before we said cephalic accounts for 30%, gastric phase accounts for 60% of it. And we have three mechanisms by which we release gastric juice in our gastric phase. We have short reflex, which is also known as local or ENS. ENS stands for enteric nervous system. I think you guys already took it, but what it is it's like your own mini nervous system, but in your stomach. Okay, so it's completely independent from the rest of the nervous system. It controls the stomach locally, just there. Then we have our long reflex, which is vagovagal. And can you read? Okay, I will do it right after this. Someone asked if you can re-explain the first clinical. I will do it right after this, after the gastric phase. So the long reflex is also called vagovagal, and then we have digestion of protein. All three of these stimulate the release of gastric acid in some way, okay? So we're going to go with the first method first. So when you have distension in your stomach, distension happens because you have food in your stomach that's stretching on the walls of the stomach and everything. So obviously there's mechanical receptors or any kind of receptors there that detect that there's food there. Also temperature, whether your food is hot or cold, that's detected by receptors there. And then also the chemicals in our food, that is also detected by receptors in our stomach. We will then have a short reflex in our stomach. This is the short reflex pathway that I'm going to be explaining. Our receptors are stimulated because of any of these stimuli, right? So after these receptors are stimulated, it will then stimulate the submucosal or Meissner's plexus in our stomach. So we have a plexus that is in between the layers of our stomach, which is part of the enteric nervous system. Okay? Meissner's plexus, it doesn't go to the central nervous system. This all is purely happening in our stomach, which is why we call it short reflex or local, because it doesn't go anywhere far away. And then the Meissner's plexus releases acetylcholine, and then we all remember what acetylcholine does. It stimulates enterochromaffin-like cells to release histamine, to then stimulate parietal cells, or it can also stimulate G cells to release gastrin, which also stimulates parietal cells and ECL cells, and it's all the cycle. And then ACH can also directly stimulate parietal cells. So just a short recap of the short reflex pathway. We have distension or any of these stimuli. It stimulates the receptors, which then stimulates the submucosal plexus, sorry, which can release acetylcholine on its own, no vagus nerve, just the plexus is releasing ACH, which then stimulates the whole pathway. Okay, that's our first pathway down. Our second pathway is the long reflex or the vagovagal reflex. This one is slightly longer because we're going all the way to the central nervous system. It's no longer locally happening in our stomach. It goes all the way to our central nervous system, sends sensory signals to the medulla in our brain, okay? And then this activates our vagus nerve. So we have afferent signals going to our medulla. It processes this in some way, and then it through efferent nerves, it activates our vagus nerve, which again, as we said, releases acetylcholine. So this is the long reflex way, goes all the way up here and then back down, and then vagus nerve releases acetylcholine. And at the same time, as we mentioned before, the vagus nerve also releases GRP, which is gastrin releasing peptide. Okay. And then again, gastrin releasing peptide can stimulate G cells. G cells release gastrin. Gastrin stimulates parietal cells. It also secretes ACH and it stimulates the whole pathway all over again. And now, last but not least, we have digestion of proteins. Digestion of proteins, we have peptides and amino acids. So whenever they enter your stomach, our stomach gets activated and it stimulates, as we mentioned before, one of the stimuli for G cells is peptides and amino acids. So in this case, this is where it happens. Our G cell notices that we have peptides and amino acids in our stomach, so it releases gastrin. So yeah, this one is pretty straightforward. Just peptides, amino acids, stimulates G cells, gastrin, stimulates the whole path. So for the person who asked if avagotomy would stop the gastric phase, it would not. Because as you can see, although the vagus nerve in the long reflex pathway would be affected, we still have the short reflex pathway, which can still release acetylcholine to stimulate all of this. And then simultaneously, we still have the digestion of proteins, which then would directly stimulate the G cell and release gastrin. So our gastric phase is all good. There's no problem with that. The only problem that would be affected if we did have a vagotomy would be the long reflex pathway. I hope that's all good. And then the last point here that I want to mention is that if the pH of the gastric juice becomes too low, like below 3.5 or anything like that, there will be a negative feedback mechanism, which will go to our brain and everything, all about negative feedback. And then it will stimulate D cells to release somatostatin. And again, as we said before, somatostatin inhibits parietal cells and G cells so that we have no more release of gastrin, less release of acid so that the pH in our stomach can rise so it doesn't become dangerous. And that's it for the gastric phase. Now I'm just going to go back really quickly because someone asked about the first clinical, so I will do that right now. The first clinical talks about a vagotomy. And what a vagotomy is removal of the vagus nerve, okay? So there's no more vagus nerve at all whatsoever. So what we do with that is because as you can see, the vagus nerve is responsible for everything in the cephalic phase. It's not responsible for everything in the gastric phase, only in the cephalic phase, because it is activated and it releases GRP and ACH. So if we don't have the vagus nerve anymore, which is what a vagotomy is, we will have none of these things, no ACH, no GRP, nothing like that. So our enterochromaffin-like cells will not be stimulated, parietal cells will not be stimulated, G cells not stimulated, no acid secretion in the cephalic phase only. Gastric and intestinal phase are fine, but in the cephalic phase, it's all gone. I hope that answered your question, or if you understand it now, if you don't, feel free to tell me. I can still try to explain it in a different way, but just let me know. Okay, so now that we know the cephalic phase and we know the cephalic phase and we know the gastric phase, we're going to go finally to the intestinal phase. Let me just go through this really quickly. Here we go, the intestinal phase. Now, as we said, the cephalic phase accounts for 30% of our gastric juice. Our gastric phase accounts for 60%, and our intestinal phase now accounts for 10%, which is very little. And the reason is because of this is because the intestinal phase, aslan, is not meant to be there for secretion of a gastric juice. Actually, it's kind of the opposite. We want it to be inhibitory. We do not want that much gastric juice anymore, okay? And how this phase works is that, first of all, if the stomach acidity falls below 2.5 or 3, which is very bad, that's actually really low for the stomach because it needs to be around 3 to 3.5, which is the perfect optimum pH for enzymes to work. If it falls below that, you're at risk of developing ulcers or just nasty side effects. Or if the chyme in our duodenum, a chyme is basically what comes out of our stomach and enters our small intestines. That's all what chyme is. If it is highly acidic, so again, this can damage the walls of our duodenum. Hypertonic. Hypertonic means very concentrated, like it's not diluted at all. It's very, very concentrated, or it has very high amino acids, monosaccharides, or fatty acids, meaning that it didn't properly get breakdown, or again, it's not diluted. Then we have to take action. And I know this part looks a little bit overwhelming, but this is the first action that we take for intestinal phase. So I repeat that this phase is mainly inhibitory. So we're trying to get rid of any acid. So this is the steps that happens through it. So first we have the hormonal mechanism, which you can remember as SICD. SICD. We have four cells that release hormones to try either bring the pH back to its normal pH or to a basic pH to protect us, or to stop the secretion of acid. So the first cell that we have that works in the hormonal mechanism is S cell, and it releases secretin. So you can just remember it as S cell for secretin. And S cells or secretin stimulates the release of pancreatic bicarbonate, okay? So that is acting like a buffer, and it also decreases motility. And what motility does, why we're trying to decrease motility, is that if we have too much motility, even more chyme will go directly into the duodenum, and it will still be messed up with low pH and high amino acids and everything bad that we don't want. So secretin tells our stomach to calm down, and it tells it to stay in the stomach a little bit longer so we can digest it or we can figure out whatever's going on. Okay, so that's for our first cell, secretin, S for S, and then we'll go to our I cells, which releases CCK. CCK decreases gastric emptying, which again has the same concept of motility. We just don't want so much to go into our duodenum while it's still very acidic or while it's still too concentrated or anything else. It also stimulates pancreatic enzymes so that we can, if it is still very concentrated or it still has very high monosaccharides or amino acids, it can still be broken down. Okay, so I cells for CCK. K cells release GIP. GIP is pretty simple. It goes directly to the parietal cells and it inhibits gastric acid secretion. And then last but not least, we have the D cells, which we already know. D cells release our somatostatin and they inhibit parietal cells, G cells, and enterochromaffin-like cells. So SIC-D, secretin, S cells for secretin, I cells for CCK, K cells for GIP, and D cells for somatostatin. I think you guys already took this in your first lesson, so I hope it's not too overwhelming, but just remember SIC-D for inhibiting mechanisms. Okay, then we go to our nervous mechanism, which also happens to inhibit this. First, we have our enteric nervous system, which, as I said again, is just locally placed nervous system. It's just there. And the enteric nervous system detects osmolarity, which is basically the same thing as what hypertonicness is or anything like that. It detects how diluted or how concentrated it is. It also detects glucose, any glucose, fat, or acidity in the intestines. If the enteric nervous system notices anything like this or notices anything out of place, it will lead to local inhibition of gastric secretion and motility. So again, we want to stop the secretion of acid so that the chyme that's coming out of our stomach is less acidic, and it also decreases motility, so less chyme is going into the duodenum. And then we also have the long reflex, so sensory signals. This is no longer in the enteric nervous system. It goes all the way up to our CNS, and then sympathetic nerves come and inhibit stomach secretion and motility. Why sympathetic nerves? Because we said before that parasympathetic is for rest and digest. So parasympathetic is everything you need for digestion, releasing acid, releasing enzymes, more motility. Sympathetic is the exact opposite. When we want to stop digestion, when we want everything to do with digestion to stop, our sympathetic nervous system activates and inhibits all of that. So again, we have two mechanisms: hormonal mechanism with SIC-D, and then nervous mechanism with enteric nervous system and long reflex. And both of these together combine and they lead to the inhibition of parietal cells and the prevention of excessive acid secretion and ulceration. So one last time, intestinal phase, we want to inhibit any of the acid secretion or the motility or enzymes. Everything stop. So we have hormonal and nervous, and that inhibits the parietal cells and excessive acid secretion so that we don't have ulceration, both in the stomach or in the duodenum. And that wraps up the intestinal phase. If it's all inhibited, where inhibitory, where does the gastric juice come from? There is some coming out still if, for example, it's all fine. Like our acid, our chyme is fine, the pH is fine, everything is fine, and they just want to digest a little bit more before it goes into the duodenum. It can release some gastric acid, but again, the number is very like it's very in the air. It's not 100%. Are the enterogastric reflexes long ones or short ones? The enterogastric reflexes, I'm pretty sure are long, but I'm not 100% sure. I could double check if you want and get back to you later, but I'm not 100% sure. Sorry. Okay, so I'm pretty sure it is long though. Okay, so with that, we're gonna go to the next checkpoint. We finished the second part, which is the acid secretory phases, and I'm gonna ask the next question. But before I do that, if anyone has any questions, you can feel free to ask me again. And with that, if there's nothing, I'll just do the next question. So, two medical students are studying for their physiology final exam and decide to take a break for lunchtime for a lunchtime hamburger. Before reaching the cafeteria, nervous impulses from the dorsal vagal complex will initiate gastric acid secretion by triggering the release of which neurotransmitter from the enteric nervous system? Norepinephrine, VIP, substance P, gastrin-releasing peptide, or nitric oxide. Okay, so we got a D, which is perfect. The answer is D. As you remember, the vagus nerve releases GRP, which then can activate all the other cells, the G cells, the parietal cells, the enterochromaffin-like cell. I mean, sorry, GRP only activates the G cells. So that's why we still have some part of the enteric nervous system. Okay. Our next part is everything about HCl. 100, it's a lot. Surprisingly, you have to know a lot about HCl, but don't worry, just go with me step by step, and inshallah, it should be fine. Okay, so our first thing that we're going to be taking is mechanism of HCl secretion by the gastric parietal cells. So what we're going to do now is how does hydrochloric acid come from a cell? How does it form? Because it sounds kind of insane. How does a cell make acid? This is the way it does. Okay, so when you have respiration in any of your cells, obviously respiration occurs and it releases carbon dioxide, okay? So the carbon dioxide that parietal cells have actually combines with water, and it does that through an enzyme called carbonic anhydrase. When CO2 combines with water, it forms a bicarbonate ion through that carbonic anhydrase. But the problem is bicarbonate is not stable at all. It's not stable. It will barely stay in your cell for like even a second. So it really quickly breaks down into a proton ion and a bicarbonate. Sorry, this is wrong, but it will break down later into a bicarbonate ion and an H plus ion, okay? This should be carbonic acid. It should be H2CO3, okay? And then it will break down into a proton and bicarbonate ion, okay? So when that happens, we have our proton ion that goes through this proton potassium pump, and it uses ATP in the process. So proton goes out, potassium goes in, okay? That's the first part of our HCl. So we have HCl. The first part comes from the breaking down of the carbonic acid into a proton. That proton goes through the proton pump and it releases H plus into the stomach lumen. So the proton and potassium pump is part of the apical surface of the cell, okay? Now, what happens to the other part? What happens to the bicarbonate ion from the carbonic acid? Well, the bicarbonate ion goes through another exchanger, which is called the bicarbonate chloride exchanger. And it's the same concept, but this time the bicarbonate is going into the blood and the chloride ion is going into the cell. So here we have the proton going into the stomach lumen, the bicarbonate ion is going into the blood in the capillaries. And a little bit of a fun fact is that when you have a meal, your blood turns slightly alkaline, and this is the reason because it's trying to produce more acid, and in the process, it releases bicarbonate into your blood, and bicarbonate is alkaline, so your blood becomes a little bit alkaline. And in the process, we have Cl entering your cell, which is perfect for us because Cl is the next thing we need to make the hydrochloric acid, which consists of HCl. So this diffuses directly, just like so, through apical membrane. It doesn't need to have a pump or any exchanging. It just directly goes. So then we have our hydrogen ion, our proton, basically, and our Cl, and they can combine perfectly to form our acid. And this is the way it does it. There's also another pump over here, which is called the sodium-potassium pump. And this is here to just maintain the ion gradient. It's not really important in the process specifically for the creation of hydrochloric acid. It's just there to maintain ion gradient. So just to recap, we have carbon dioxide from respiration, and we have carbon, and we have water. This combines in the presence of carbonic acid to make carbonic acid. Carbonic acid not stable at all. It breaks down into the proton and a bicarbonate ion. Our proton goes outside to our stomach lumen, and in the process of ATP, it also brings a potassium ion in. On the other end, a bicarbonate ion goes into the blood in our capillaries, and in the process brings in a chloride ion. Okay, chloride ion coming in is perfect because we want it to diffuse out to combine with our H plus to make hydrochloric acid. In the process or in the background, we have a sodium-potassium pump that's just working to maintain the gradient of ions in our cell. And that's how you make hydrochloric acid. Okay, now that we know how we make hydrochloric acid, how is it regulated? How do we decide what comes in and what goes out? So this picture might look a little bit overwhelming. I know it did look overwhelming to me when I first studied it, so inshallah, I'll make it as simple as possible. Think of it as the parietal cells have 3 on signals and 2 off signals, okay? 3 on signals stimulates our parietal cells, which is perfect. And then the 2 off signals, which we'll see later, inhibits it. Our first on signal is the acetylcholine. And the acetylcholine comes from our vagus nerve. So you can see it right here. Vagus nerve releases ACH, and then the ACH will bind to our M3 receptor, or our muscarinic receptor that we mentioned earlier, okay? And that will activate the GQ pathway. And what the GQ pathway is basically a cell signaling cascade. So it just goes to second messengers and then things like that. It's a very long process. I'm pretty sure you guys took it in the cell signaling lectures in mole, but what you need to know for this one right now is that it activates the GQ pathway as one of the G proteins, which then causes IP3 and calcium to increase. Again, this is just the pathway for GQ. This is something you just have to memorize. There's not much understanding behind it. And then when we release IP3 and calcium, that will then stimulate the activity of the proton potassium pump, and then more protons will be released, just like here. So basically what it does is that IP3 and calcium acts on this channel right here, so it moves faster and releases more protons. That's basically it. That's for acetylcholine. Then we have gastrin, which has a different receptor, but other than that, everything is basically the exact same. Gastrin binds to a different receptor, not the M3 receptor, but in this case, the CCKB receptor. But then that activates the GQ pathway. GQ pathway activates IP3 and calcium. And then IP3 and calcium activate the proton potassium pump. So again, more protons will be pumped out, more acid secretion, more hydrochloric acid, everything like that. That's for ACH and gastrin. Just remember they have the same GQ, only the receptors are different, which is M3 and CCKB. And then last but not least, for our last on signal, we have histamine. Histamine binds to an H2 receptor. So this one is different, and it also stimulates a different G protein pathway. So before we had GQ to IP3 and calcium, now we have GS. GS is a pathway which, when it's activated, it activates an enzyme called adenyl cyclase, okay? Adenyl cyclase is an enzyme which can then increase cyclic AMP. So just to repeat, GS is activated, that activates the enzyme adenyl cyclase. Adenyl cyclase increases cyclic AMP, and then cyclic AMP can act on our proton potassium pump. Okay, so we have 2 things acting on our proton potassium pump to increase it. We have IP3 or calcium, and then we have cyclic AMP. So a quick recap of everything we have: ACH, M3, GQ, gastrin, CCKB, GQ, and then histamine, H2, GS. Okay, I hope that's all clear. we're going to now move on to there are two off signals, which we have somatostatin and prostaglandins. So for this one, we don't really have a specific receptor for it. We just have the pathway that it uses. In this case, we have GI. Now, if you look at these two, it's pretty like you can use this as a way to memorize. The S in GS, you can think of it as stimulation, and then the I in GI, inhibitory, right? So in this case, we're using somatostatin to activate the inhibitory pathway. So the inhibitory pathway will, unlike the stimulatory pathway, this one will stop our adenyl cyclase. So before, adenyl cyclase was what we needed to increase cyclic AMP to activate our proton potassium pump. But in this case, we're activating GI, which decreases our adenyl cyclase. So we're going to have a decrease in cyclic AMP, and then thus our potassium proton pump will work a lot less, so less acid secretion, everything. And as we said earlier, somatostatin is released from our D cells, it decreases acid and everything and whatnot. So this makes sense. And then we also have prostaglandins. Prostaglandins also work in the same pathway. It's the exact same, nothing different from somatostatins. It activates GI. GI decreases cyclic AMP. Cyclic AMP, then if there's less cyclic AMP, less of the pump will be activated and thus no more acid secretion or less acid secretion. So I hope that makes sense. Somatostatin and prostaglandins both activate GI. And if GI is activated, it will decrease the adenyl cyclase and the cyclic AMP. If any of you have any questions, please feel free to ask me. So now you might be seeing some weird names every here and there, omeprazole and then atropine and then cimetidine. We already mentioned atropine earlier as the muscarinic blocker or the muscarinic receptor blocker, and it does the same thing that I asked. It's going to block our M3 receptor so that ACH can no longer bind. So this is a way that we can block it and decrease the acid secretion if we need it. For example, if there's a condition where we want to stop acid secretion to stop ulcers or anything like that, atropine could be a way. Same thing with cimetidine. It's a histamine blocker, so it blocks the H2 receptor so that we can have less GS, less cyclic AMP, and there's less histamine to stimulate the parietal cells. And then last but not least, we have omeprazole, which is a proton pump inhibitor. And its name is basically what it is. It's a proton pump inhibitor. So we have none, this is not working. I just want you to differentiate the fact that omeprazole acts at the very last step, which is the proton pump, whereas atropine works earlier. So atropine would not completely stop acid secretion, and cimetidine would not completely stop acid secretion. But omeprazole, while it might not stop it completely, it does a lot more than atropine and cimetidine, which is why when you see cases of gut ulcer diseases or things that are really severe, they always prescribe proton pump inhibitors because it's the stronger option and it helps protect the stomach more. Okay, so now that we have finished finally what the regulation of acid, we're going to be talking about the activation of pepsin, which is pretty straightforward, pretty simple. So first thing I want you to note about pepsin is that if the pH is less than 3.5, the pepsin will inactivate. If the pH is more than 3.5, sorry, it will inactivate the pepsin. So pepsin will no longer be effective at a pH higher than 3.5. That's because it needs to be very low for it to no longer be in its zymogen form. So here we have pepsinogen, okay? And pepsinogen is what comes out of our chief cell. So as we mentioned earlier, our chief cell releases gastric lipase and pepsinogen. Pepsinogen is just a zymogen, and what a zymogen is that it's an inactive enzyme, so it's not working right now. We need to put it in the correct condition so that it will become its enzyme, okay? So right now, let's say, for example, our pH in our stomach is 3 to 5, okay? Pepsinogen will slowly start converting to pepsin. And the reason why I say slowly is because at pH that is closer to 5, 4.5, 4 in that region is low. Obviously, it's low, but it's not low enough to quickly activate it, okay? So at this pH, we'll have active enzyme pepsin. It will still be there. But if we have a pH less than 3, that's when we have rapid and fast activation of pepsinogen. So basically, we just want pepsinogen to turn into pepsin so that we can digest food. If it's still in its pepsinogen, no food will be digested. None of the proteins will be there. It'll just stay stuck there on the chief cell. And now that we know one function of hydrochloric acid, which is creating the optimum pH for pepsinogen to be converted into pepsin, we can talk about a few of the other ones. The first one, creating acidic medium for pepsinogen. Second one is protein denaturation. So this is just plays a role in breaking down food. We want to denature proteins in our food so that it's no longer huge chunks. It's also antibacterial. So we can stop the bacteria from flourishing in our stomach, and we're going to be talking about that a lot more. There's like a whole part for bacteria in our stomach. It also helps with iron absorption, and it helps with motor and secretory regulation of the gastrointestinal tract. So it also tells us how much more secretion should we have, or how much more motility should we have in our stomach, and that's basically what it's telling us with HCl. So it's very useful. Okay, now we're going to be talking about the protective and damaging factors on the mucosa. So obviously HCl is good for many number of reasons, but if it becomes too much, if the environment becomes too acidic, it can also be very dangerous. Again, because of ulcers, because of damage, you have a hole, no one wants a hole in your stomach. So that's why it's pretty bad for you. So we have mechanisms for our stomach to protect itself, and there are also some things that damage our stomach, right? So first we'll talk about the protective factors of our stomach, which is, as we mentioned almost a million times before, is the mucus and the bicarbonate ion. Mucus acts as a physical barrier. It stops acid from directly touching the wall of our stomach and creating a hole. And then the bicarbonate acts as a buffer, so it fights against any acid in our stomach and it basically protects it. Prostaglandins, we mentioned this earlier when we were talking about the regulation of secretions. This is one of the inhibitory that activates GI, and then GI decreases cyclic AMP. And then when cyclic AMP decreases, it will decrease the activity of our proton pumps. So that's why prostaglandins also protect us. Also, good mucosal blood flow also protects us, because if we have good mucosal blood flow, our cells will not be ischemic, or aka it will not lack oxygen. So more regeneration, more renewal. Our cells can be healthy, and it won't, the acid will not be easily able to penetrate through it, if that makes sense. And then last but not least, we have tight junctions. Tight junctions in between is pretty straightforward. It prevents any of the acid from leaking between the cells and causing even bigger gaps. And then for damaging factors that are present in our stomach, we have the very obvious thing, hydrochloric acid. We also have pepsin. Because pepsin is an enzyme and our stomach is essentially a muscle, and muscles are made of proteins, if pepsin touches the protein stomach, that's very bad. Obviously, you can tell. So pepsin could digest our own stomach, which is very scary. So, and then we also have H. pylori. H. pylori is also called Heliobacter pylori, and that is a type of bacterium. And this can cause a condition which is very bad. We'll be talking about it more. So let's just keep that aside for now. We have NSAIDs, which are non-steroidal anti-inflammatory drugs. This is like ibuprofen or any of these painkillers that you get at the store. These are bad because you're going to learn this next year and more upon it more. But just in case, if you're curious, NSAIDs stop the release of prostaglandins. And as you can see over here, prostaglandins is a protective factor. So if we have less prostaglandins, then acid will not be as inhibited. It will stop, more acid will be secreted because prostaglandins inhibits acid secretion. So with more NSAIDs means less prostaglandins means more acid secretion. And then alcohol, smoking, stress, all of these also play a role on the regulation of your prostaglandins and liver function and things like that, which then also cause damage to your stomach. Okay, now, as I said earlier, we're going to be talking a lot more about H. pylori, and this is where it comes into factor. So H. pylori is a bacteria, and it's basically a digestive system, a gastrointestinal bacteria. So its main place where it likes to reside is in your gastrointestinal system. And it's basically when you eat any bad food, for example, or you're at a specific area where it's more prevalent, this is how you can get H. pylori in your stomach. So H. pylori is normally there But the problem with H. pylori is that it has an enzyme called urease. And urease is a problem. It's a huge problem because urease can create ammonia. And when we have ammonia, ammonia is alkaline. So imagine you have an acidic environment, and that acidic environment is there to protect you from bacteria because it can kill the bacteria, right? But if we make ammonia, the environment will become alkaline. So the bacteria can live because it's no longer acidic for it. So it can live, it can flourish, it can become everything it needs to become. And the environment is alkaline, so it can spread throughout the entire stomach. It's a huge problem, right? So this H. pylori can then also spread to your duodenum, and it can also infect your duodenum, and then there it causes inflammation and so much bad things in your duodenum. And then because of that inflammation, there's going to be less bicarbonate secretion because everything in your duodenum is messed up. So there's less bicarbonate, so less buffering, stopping the acid from being secreted. It's a huge problem. And then on the other side, H. pylori can then also decrease the release of somatostatin from D cells. So as we said earlier, D cells release somatostatin. Somatostatin helps inhibit G cells and our parietal cells. But when we have a H. pylori infection, it actually affects our D cells. So our D cells cannot secretes as much as somatostatin, okay? And then more gastrin is released because we have less somatostatin. And then more acid is released because we have more gastrin. So it's just more acid, so acidic. We have less bicarbonate, so we have less protection, and it's a huge problem, which then in the end can lead to a gastric ulcer or a duodenal ulcer because we have less protective mechanisms because of the bacteria just messing up our environment totally. So a quick recap, H. pylori has urease. Urease makes ammonia. So then the H. pylori can live more. It can cause inflammation in the duodenum to release more bicarbonate, or it can cause decreased somatostatin, and that means no more inhibition of acid secretion. So then there's more gastrin release, more H plus secretion, and then ulcers. Very sad. Now we'll talk about peptic ulcer disease, which can happen because of a H. pylori infection, right? So in this photo, you can see that there's holes in the stomach wall and in the duodenum wall. And gastric ulcers usually arise because there's a defective mucosal barrier. So if it's in the stomach region, it's usually because we have, for example, less mucus secretion, or we have less bicarbonate secretion, so that makes more holes in the stomach. For duodenal ulcers, it's usually because there's excess H plus secretion because the main thing that duodenum does is that it secretes a lot of mucus and it secretes a lot of HCO3 or bicarbonate. So it's a lot less likely that the problem would be the mucosal barrier. It's more likely that there's excess H plus secretion, which makes the chyme very acidic and it makes walls in our duodenum, right? So there is a lot of causes for what could happen with peptic ulcer disease. One of them being excessive acid secretion. And a syndrome that causes excess acid secretion is called Zollinger-Ellison syndrome, which increases the secretion of gastrin acting on the CCKB receptor. Okay, so if we have more gastrin, more CCKB will be activated on the G cells, and then more gastrin, more acting on parietal cells, more acid. That's one of the things. Or it could be decrease in the protective properties from the mucosal barrier, like in H. pylori. As we mentioned here before, H. pylori causes decrease in the bicarbonate secretion, so that damages our mucosal barrier, unfortunately. So that can be one of the causes. Other causes of peptic ulcer disease is because of gastritis, reduced blood flow, which leads to ischemia, smoking, alcohol, NSAIDs, and inflammation. As I mentioned earlier, NSAIDs because it reduces the prostaglandins, smoking, and alcohol, and inflammation because it's stress, and then stress leads to more ulcers. And then gastritis basically just means inflammation of our stomach, so same thing. And then reduced blood flow reduces the regenerative capability of our cells so that they're not as healthy as before and they easily become more damaged by pHs that wouldn't normally damage our stomach. And the treatments for this is one, it can be an antibiotic, and this is in the case if the reason for your peptic ulcer is H. pylori, because H. pylori is a bacteria. So if you use antibiotic, it could decrease H. pylori so that your protective property of your mucosal barrier will be better again. And then you can also have PPIs, proton pump inhibitors. Again, as the name indicates, it just stops the proton pumps from secreting protons, so it's safer for you and your stomach and everything. And then we have inhibitors of gastric secretions and selective vagotomy. So by inhibitors of gastric secretions, we mean the ones that we mentioned earlier, like atropine, which is a muscarinic receptor blocker, or even cimetidine, which is a histamine receptor blocker. And then selective vagotomy, this is when we don't like for a full vagotomy is when we get rid of all of the vagus nerve, but for a selective vagotomy, it's just part of the vagus nerve so that there's still secretion, but not as much. And then again, this is just a repeat of what I said earlier. NSAIDs like ibuprofen damages the mucosal barrier in the stomach and the duodenum because they inhibit prostaglandins. And prostaglandins inhibits the release of acid. So that's why NSAIDs are pretty dangerous. So don't just take ibuprofen or anything every single day because they can cause actual serious damage to your stomach and it may lead to something really dangerous. And then again, as I said earlier, antibiotics relieve peptic ulcer disease because they kill the pathogens, aka H. pylori, which damages the mucosal barrier. Okay, so finally, we finished with everything about hydrochloric acid or about H plus, everything about that. So we have our third checkpoint. So if you have any questions, feel free to ask. If not, I'll just bring up the question. A 68-year-old woman with hematemesis has heartburn and stomach pain. Endoscopy shows inflammation involving the gastric body and antrum, as well as a small gastric ulcer. Biopsies were positive for Helicobacter pylori. H. pylori damages the gastric mucosa primarily by increasing mucosal levels of which substance? A. Ammonium, B. Bile salts, C. Gastrin, D. NSAIDs, and E. Pepsin. Okay, amazing. Everyone's saying A, which is 100% correct. As we said earlier, H. pylori releases urease, and then urease makes ammonia or ammonium, which creates the alkaline environment so that the H. pylori can persevere and survive and cause tons of damage. Okay, everything's going well, I hope. If you have any questions, again, feel free to ask. And finally, we're on our last section, which is gastric motility and emptying. Okay, so first, what we're going to talk about is the sphincters in the GI, which is our lower esophageal sphincter and our pylorus, okay? So GI sphincters are tonically contracted. So our lower esophageal sphincter and our pylorus are usually closed all the time. Tonically just means all the time closed. So they are normally closed, and the inhibitory motor neurons are usually inactive. What the inhibitory motor neurons are, it's just they inhibit contraction, basically. So you can see it in the photos here, inhibitory motor neurons, inhibitory motor neurons. They are usually inactive because we usually want the sphincters to be closed. One reason because we want the lower esophageal sphincter to be closed because we don't want reflux, and then we want the pylorus to be closed because we don't want too much food to go into the duodenum and overwhelm it. So they are usually closed, and the inhibitory motor neurons are usually inactive. However, when they are activated, for example, when we have something going through our esophagus and we want to open our lower esophageal sphincter so that the food can enter our stomach, our inhibitory motor neurons release VIP and release nitric oxide. And these two act on our sphincters so that they open and they relax so that it can go through. So again, inhibitory motor neurons, when they are activated, for example, during swallowing or anything like that, we have release of VIP or NO, nitric oxide, which relaxes our two sphincters, which you can see here. Here, probably VIP and nitric oxide are being released, and then they're open for anything to go through. So the lower esophageal sphincter, as I mentioned earlier, only opens during swallowing, and then our pylorus is regulated by chyme composition. For example, if the chyme is very acidic or too hypertonic or there's so much fat in it, our sphincter will close again. Our pylorus will close again because we don't want that in our duodenum. We don't accept that in our duodenum. We only let it in if it's less acidic, less hyperosmotic, or less fatty. But if it was in good state and if it's ready to go into our duodenum, our inhibitory motor neurons again will release VIP and nitric oxide to open the pylorus, and then it can pass through easily. Okay, so that's for our Would histamine be correct there? I don't think histamine would because I think histamine is just, oh, sorry, are you asking about the previous question? I think, would histamine be correct here? Biopsies were positive for H. pylori. I don't think it would be correct because actually it would probably be correct, actually. You're right, because it does stimulate the cells in our what's it called? Stomach. For example, the somatostatin will decrease, so more gastrin, and then gastrin can act on enterochromaffin-like cells. So histamine, if it was an option, it would probably most likely be correct. You're right. Okay. Um, For the next part, now that we're past that, we'll be talking about the pacemaking cells of the GIT. Uh, Now, for this, I want to emphasize on one thing. The membrane potential of our stomach is always fluctuating. It's never constant, okay? And because of that, we have slow waves in our stomach, which is also called basal electric rhythm. So just imagine the membrane potential of the pacemaker cells just going up and down and up and down. This does not mean that they're contracting. This is not a contraction. It's just like small fluctuations of our stomach. So it's never still, it's just slowly fluctuating, like waves. So it's never completely at rest because it has rhythmic fluctuation called the basal electric rhythm or slow waves. And then these slow waves are generated by interstitial cells of Kegel, ICC. So these are what we call interstitial cells of Kegel. They are the things that have the fluctuating sorry, have the fluctuating membrane potential, okay? And these interstitial cells of Kegel are found between the longitudinal and circular muscular layers, closer to the Auerbach's plexus, okay? So they are just in between the longitudinal and circular layers, and they cause the slow fluctuation and the waves to go through the stomach. They are spread through the gap junctions. So these slow waves, there are gap junctions in between the interstitial cells in these areas, and they are spread through that. And if these slow waves, so as I said before, the slow waves are just slowly fluctuating. But if a stimulus causes it to contract and the slow waves suddenly increase and it crosses the threshold potential is what you need to pass in order to create an action potential. If that happens, then a contraction occurs. But normally it's not contraction. Normally it's just slow wave like that. Okay, so that's it for the slow waves. You don't really need to remember these frequencies. This is just for there. I'm pretty sure there's no way she's gonna ask anything about that. But just for your information, in the stomach, the slow waves are usually 3 to 5 per minute. For the small intestine, 12 to 20, and then for the colon, 6 to 8. Again, I don't expect her to ask this. Okay, so now that we know what the slow waves are, just to recap, they are slow fluctuations in the membrane potential that causes the stomach to slowly just have some small waves go through it. We can talk about this. Okay, so what are we looking at here? Let's start with when we're at rest. When we're at rest, we have small muscle tone. So our membrane potential, as we said earlier, is fluctuating a little bit, but there's no contractions at the moment. So it doesn't reach threshold because there's no stimulus causing it to reach threshold. There's nothing to make an action potential and to lead to contractions. So we have very minimal muscle tone. There's no muscle tone. It's just very like it's there, but it's not very strong. There's no strong contractions. It's just there, you know? If we have it stimulated, for example, we want the muscle to contract a lot, to contract, then the membrane potential will be stimulated to go past the threshold potential. And again, remember that the threshold potential is what we need to make it action potentials. If it makes the action potential, then we can have contractions. So, for example, what can stimulate the stomach is ACH from our vagus nerve again, stretch. If there's food, it causes distension and everything. And then hormones from our cells in our stomach, like the G cells, the D cells, and everything. So slow wave amplitude increases and then it reaches threshold, which is what we have here. It's increasing. You can assume that the threshold potential is somewhere over here. So then here, the muscle tone is a lot larger from here because we reached the threshold. So that makes the calcium ions to open, more a spike potential happens, and it leads to smooth muscle contraction. And one thing I want you to know about this part or in the stimulated part is that if you have more spikes, so more increases past the threshold potential, and the membrane potential has a higher amplitude. That means a stronger contraction. So if it's less stimulated, if it's somewhere here, it would like the amplitude was shorter, it would not be as strong as a contraction. But for example, here it's probably really strong. And because it's a lot, here you can only see 4 waves, but here you can see 5 waves. That means the frequency is higher, so the contraction would probably be stronger here as well. And then last but not least, if it's inhibited, so we don't want any contraction at all. This is for example, especially in the places of the sphincters, this is when we have inhibitory input. So for example, VIP, nitric acid or sympathetic stimulation, when we have inhibitory neurons releasing these, it completely stops any muscle tone whatsoever. So there's not even the fluctuations. So when we want to open the sphincters, this is pretty much what it looks like. It's just slow waves that are below the potential. It doesn't reach the threshold potential, so there's no contraction, small contraction. That's why our sphincters are completely relaxed when we're trying to bring food through it. It's because of we have these inhibitory input. So everything is relaxed, everything is chill, everything is fine. So at rest, normally our stomach is like this. If it's stimulated through ACH or anything stretch hormone, it will have more spikes with higher amplitude, leading to more contractions. And when we're talking about sphincters, they usually have input with VIP and NO, which causes the membrane to be completely hyperpolarized. So it has no chance whatsoever to reach the threshold, and so it doesn't contract. It just remains relaxed. Okay, now that we're done with that, we can talk about adaptive relaxation. What adaptive relaxation is, it allows the stomach to expand and accommodate food without increasing too much of the gastric pressure. Because if you ate and ate and ate and ate and you kept eating and the stomach just stayed as it is, then it would be a big problem. It would basically explode. We don't want so much pressure building up in our stomach that it causes a problem. So this is what we have in the adaptive relaxation mechanism, which helps us prevent too much increase of the pressure inside our stomach. So gastric stretch receptors in our stomach, they detect that, wait, she's eating more. This person is eating more. Our stomach is having more pressure in it. We need something to happen. So it sends vagal efferents. Efferents is basically just the sensory signals. They send it to the medulla, and then the medulla will detect that and it will send efferents to the ENS, the enteric nervous system, which is locally there in our stomach. This causes interneuronal circuits to do their things, and it stimulates the inhibitory motor neurons. So just to recap, stretch in our stomach stimulates the gastric stretch receptors. This goes all the way to our brain. Brain sends signals through the efferents to the enteric nervous system to activate the inhibitory motor neurons. And as we mentioned many, many, many times before, inhibitory motor neurons release VIP and nitric oxide to cause muscle relaxation. So when we have that muscle relaxation, the stomach can more easily expand because it's not as contracted, and so it can accommodate more food, and so there's less pressure buildup. Okay, so just to recap, stretch to the medulla, inhibitory motor neurons activated, muscle relaxation, more food can be accommodated. This is what we call adaptive relaxation. There's also 2 other relaxation reflexes that is in our gastrointestinal system. I mentioned the first at the very beginning of the session, which is called receptive relaxation, which is triggered by swallowing, and it allows the stomach to release food, to receive food. So when we have it in our esophagus, it's coming down, the inhibitory neurons activate and they release VIP and nitric oxide so that our lower esophageal sphincter and the cardia of our stomach relaxes so that food can easily come in. That's basically what receptive relaxation is. And then feedback relaxation is triggered by the nutrients in the small intestine, and it's the same thing. But in the case of our small intestine or in our duodenum, it allows the pylorus to be relaxed and it allows parts of our small intestine to be relaxed so the food can more easily pass through. Okay, now we have tonic contractions. Gastric tone is controlled by moment by the enteric nervous system. So basically, we have a dilemma. You could say, I think, a dilemma in our stomach because we want it to have enough contraction so that we can push contents to the antral pump. Because at the end of the day, we want our food to go into the duodenum and into the small intestine to everything. We need that. So we need to have enough pressure in our stomach and enough contraction to move the food that would normally be here to our antral pump so that it can go into our duodenum. But simultaneously, we don't want so much pressure that it leads to too high intragastric pressure and it leads to things like bloating and epigastric pain and nausea because this is not good. It's obviously uncomfortable. So our stomach has to balance between the two of those. So we have to figure out how to do this. And this is balanced by the ENS neurons or the enteric nervous system neurons. So if in that moment we think, okay, it's more important that we decrease the pressure, we don't want as much pressure, we activate our inhibitory motor neurons, which releases VIP and nitric oxide, so it decreases tone, increases volume, so it becomes bigger, like this dotted line. This is what happens if the inhibitory motor neurons are activated. It becomes the size of this dotted line, so it can expand and it can become bigger, and so our stomach can accommodate more food. On the other hand, if it was more important in that moment for the food to go directly to the antral pump and come to this region, then we would activate the excitatory motor neurons, which would release ACH and things like that, so that there's more contraction, more tone, so it becomes smaller, and now it's this dotted line on my cursor, so that it can push the food that would be in the body to the antral pump so that it can go there. So the volume is decreased and there is more pressure in the stomach, so it can go there. So again, we want to find a balance between the two. Excitatory increases pressure, decreases volume. Inhibitory decreases pressure, increases volume. Okay, so tonic contractions usually occur in this area. So this area is usually very contracted all the time and it acts as a storage. And the phasic rhythmic contractions usually occur down here. And what we mean by phasic is it just contracts and then relaxes, contracts and then relaxes, contracts and then relaxes. This is so that the food can mix more, so that there can be more propulsion towards the antral pump. It's kind of the same, a similar concept to peristalsis in our esophagus, but here it's in our stomach. We want to push the food more towards our antral pump. So here there's more phasic or rhythmic contractions. Okay, now we're going to be talking about retropulsion mixing in the stomach. It's kind of similar to what I just mentioned earlier. It happens in the distal stomach. As I mentioned here, there's basal rhythmic contractions that happen in the distal stomach or the posterior stomach, as I wrote here. So here in the distal stomach, there's a peristaltic wave that pushes food towards the pylorus. Okay, so let's imagine there's a wave going along the stomach here and it's pushing the food towards the pylorus. The pylorus closes if the food chunks are too big or too acidic, and it brings it back. So it's basically like ping pong, you could say. Peristalsis is one racket, it pushes it towards the pylorus. The pylorus is the other racket, it pushes it back. So it's kind of a back and forth. And this back and forth is called retropulsion. And our stomach naturally does that because it wants to mix and grind food more. So it's a pretty simple concept of retropulsion mixing. It's when peristalsis pushes food towards the pylorus, but the closed pylorus pushes the food back, and it's a back and forth like a tennis game, and it just causes the food to mix more and more and more. Okay, and the strength is regulated by ACH and substance P. Now we're talking about motility rate in the GIT. We're getting towards the very end of this lecture. The first thing that I want you to look at here is we have three different regions in the GIT, okay? We have the stomach, the duodenum, and the large intestine. Sorry, in the large intestine. So the only thing I want you to know here and what the MCQs love asking is which area has the fastest emptying. And as you can see here, I want you to follow these two lines, for example. The solid line, as you can see, is liquid and the dashed line is solid. So here you can see that the stomach empties fairly quickly, especially with liquid meals. So that's our first point. Liquid meals are emptied the fastest. Solid meals empty slower. So just keep that in your mind. It's very commonly asked. Liquid meals empty faster, solid not as fast. Our second point that I want to point out is that the stomach empties a lot faster than the other two. So as you can see here, liquid empties in 4 hours and solid empties at around 5 hours. But if you look at the duodenum, even though the the content and everything decreases and decreases and fluctuates, the emptying only happens after 6 and a little bit more than 6 hours. Whereas here, for the liquid, it did it in 4 hours. And then here it's even longer. It empties in 10 hours. So it's a lot. So for this point, what I want you to know from this slide is that stomach has fastest emptying and liquid empties faster. That's what basically all you need to know for this slide. And this is again what I talked about, the solid liquid phases of the stomach. It's important that for our stomach that we don't give so much solid meals to our duodenum so that it doesn't overload the intestines, okay? So our stomach controls its motility so that it can control how much of the food goes into our duodenum. But for liquid, it's just liquid. So it can very easily go through the pylorus and bypass the pylorus. So for example, if we don't want food to go into the duodenum, let's say this is the pylorus, solid food cannot go past because it's solid, it can't. But duodenum is liquid is liquid, so it can very easily pass the pylorus and go into the duodenum. And you can see this very easily here. Here we have the liquid phase, the semi-solid meal, and the solid meal. Here we have a lag phase. The liquid meal does not have a lag phase. Why? Because it's not stopped by the pylorus. It can easily slip between the cracks and do whatever it wants, and it can reach the duodenum very easily. Whereas a semi-solid meal and a solid meal would be prohibited by the pylorus to enter. So that's why semi-solid meals and solid meals stay in the stomach for longer, and they have what you call a lag phase, whereas liquid meals go directly. So I'll just read this part out. An important function of gastric motility is that the orderly delivery of chyme is at a rate that it doesn't overload the intestines. With a mixed meal in the stomach, liquids empty faster than solids because the lag phase is the time required for the grinding action of the antral pump to reduce the appropriate particle size to enter the duodenum. So we're basically just trying to make the solids appropriate particle size so that it can pass the pylorus and enter the duodenum. Now, for factors that are regulating gastric emptying, what speeds up gastric emptying is that if there's a lot of food, it's a lot of distension, so more activation, more motion, more motility, more likely for the pylorus to open and allow solids or anything to go through. And then, of course, relaxing pylorus allows gastric emptying. And then what slows down gastric emptying is what we mentioned earlier. If chyme is too acidic, hyperosmolar or fatty/protein rich, then it would cause gastric emptying to stop because as we mentioned earlier, we do not want anything that's too acidic to come into our duodenum or else it would damage it, or anything fatty or protein rich. It needs to be broken down more in order to be satisfactory for our intestines. So we want to close the pylorus and to prevent gastric emptying from happening. So this happens through the anterior gastric reflex, which activates it so that the pyloric sphincter is tightened and antral pumping decreases. So enterogastric reflex occurs, so pyloric sphincter tightens and that there's less gastric emptying, so no solid food can pass the pylorus. And then also the sympathetic nerves inhibit secretion and inhibit motility. So again, as I said before, when we don't want rest and digest, when we don't want gastric emptying, when we want nothing to do with the digestive system, we activate the sympathetic nerve so that there's less secretion, less emptying, less motility. Again, what I want you to know most importantly from the previous slide is that liquids have the fastest gastric emptying, and stomach is where gastric emptying happens the quickest. Okay, now for our very last slide, finally, it's for the vomiting reflex. Now, I really want you guys to pay attention to the order of the vomiting reflex because honestly, back when I was in first year, I thought it was unimportant because it was the last slide and I was tired of this lecture and everything, but then it came as an MCQ. So it is pretty important. So just stay with me. It's not too hard and it's pretty logical when you pay attention to it. So what happens is that the upper digestive tract becomes irritated or overdistended or overexcitable. So irritated because maybe you ate some bad food, it has some bacteria in it or something that you don't want to be in your food typically. So that's when it becomes irritated. Overly distended, for example, you eat way too much food and then you feel like vomiting, or overly excitable, which is when the stomach contracts to get rid of that food. and then disaster happens. So this causes signals to go to the vomiting center in your medulla, which is part of your brain. So just know that there's an area in your brain called the medulla, and in the medulla, there's an area called the vomiting center. This vomiting center receives signals from afferent nerves, okay? And when they receive those signals, it then sends this vomiting center sends signals through motor impulses through these nerves, the cranial nerves. I'm pretty sure you won't be asked about this, but I just added it for the sake of completion. The nerves coming from the vomiting center are 5, 7, 9, 10, and 12. 5, 7, 9, 10, 12. It's just there for completion. I don't think you will be asked about that. Now, this is the part where I think you could be asked about, which is the sequence of what happens after all of this happens. So just to recap, afferents go to the vomiting center in the medulla, and then the medulla sends motor impulses through the cranial nerves, and then the cranial nerves causes all these actions to happen. So the first thing that happens is that you take a deep breath. When you're about to vomit, you take a deep breath. So the hyoid bone, which is a bone that's around here, and the larynx rises. So our hyoid bone and our larynx rises after we take a deep breath so that our upper esophageal sphincter opens. Because obviously it comes through your esophagus and comes throughout your mouth. So obviously your upper esophageal sphincter needs to open, okay? Then the glottis closes. We close the glottis because when we're vomiting, we do not want the vomit to enter our respiratory system, which could be a huge problem. It's very bad. So obviously the glottis will close. And then we have the soft palate, which lifts and it closes the posterior nares so that it doesn't reach to your nose and comes out of your nose or anything like that. So that's very important as well. So three things happen at this point: opening of upper esophageal sphincter, closing of the glottis, and closing of the posterior nares through the soft palate lifting. Then after that, once we have all of these passageways either open so that the vomit can go directly through it, or closed so that vomit doesn't go in areas where we don't want it to be, we have strong contraction of our diaphragm and our abdominal wall muscles to increase pressure. Because obviously we need something to squeeze the stomach so that everything, its contents, goes up and the pressure raises and it can go through your esophagus. So we have strong contraction of our diaphragm over here under your lungs and the abdominal wall muscles. And then last but not least, this causes the lower esophageal sphincter to relax and expulsion of contents comes out. So just a quick recap, our GI tract becomes irritated. It activates the vomiting center of the medulla. This sends motor impulses. So our hyoid bone and our larynx open so that our upper esophageal sphincter is open. Our glottis closes so that no vomit goes into our respiratory system. Our soft palate opens so that it can block our posterior nares, so none of it goes into our nose area. And then our diaphragm contracts, our abdominal muscles contract to cause increase in pressure. And then the lower esophageal sphincter is the cherry on top. It opens, it's a direct path out, and it can go through it 100%. Finally, we are done with our final section, and we have our last checkpoint. If any of you have any questions about any of the gastric motility or any of the vomiting at all, feel free to ask me. And if not, we'll ask the last question, which is a patient with diabetes mellitus presents with gastroesophageal reflux disease accompanying gastroparesis. Which of the following statements about gastric emptying is correct? A, solids empty more rapidly than liquids. B, meals containing fat empty faster than carbohydrate-rich food. Hyperosmolality of duodenal contents initiates a decrease in gastric emptying. D, acidification of antrum increases gastric emptying, and then E, vagal stimulation decreases receptive relaxation in the upper portion of the stomach. Okay. Yes, the answer is C. Great job. C, because we want there to be osmotic balance so that there is not, yes, C. We want there to be it not to be too concentrated. We want it to digest more in the stomach so that it can break down more, so that it's more diluted and everything before it goes into the duodenum. So we do not want gastric emptying in the case of hyperosmolality. And that wraps up my session. Thank you so much. I wish you the best of luck in your TBL. Inshallah, it will go amazing and I hope you benefited from this session. And here's my contact information, here's my number, and here's my email. If you have any questions, feel free to do so. And here's the feedback form to say whatever you would like to say. And yeah, that's it. Wayaki, no problem. Thank you so much for joining, Hammad.